Yeah, I'll make you a host, Jason.
Joe, it's Mike. You've gone live.
Okay, thanks, Mike. Hey, Jason, would you mind recording since I made you the host?
Absolutely, happy to do that.
Perfect. Thank you.
What did you have?
All right, good afternoon, everyone. I think we'll go ahead and get started. We have a few folks who maybe will join us in a moment or two, but welcome to our seventh meeting of the Science Subcommittee of Governor Lamont's COVID-19 Vaccine Advisory Group. Thanks to our members for making time on a Saturday, on a dreary Saturday to join us, and thanks to anyone who may be watching us from the public or the media. We are, I think, streaming on YouTube this weekend, and we're also recording this as well. So welcome anyone who may be listening in to our meeting today. Our agenda for today is, after just a brief reminder about our meeting minutes and the role that plays in our committee's work. We'll have some brief updates on the vaccination program in general. I suspect familiar to this group. And then we have two primary items on our agenda. First, David will give us an update, as we do every month, on the latest information we have about the safety and efficacy of the currently authorized and in use vaccines. Just take us through some of the more recent information and evidence that's been published about those vaccines. And then our primary item, the reason we're meeting here today on the Saturday, is of course to discuss the Janssen or Janssen or Johnson & Johnson, three names, different pronunciations, but all the same vaccine candidate that as you are aware, is nearing an emergency use authorization. had a 22-0 favorable recommendation from VRBPAC yesterday. An EUA has been signaled from the FDA as expected probably later today. And then we'll have recommendations from the ACIP coming in the coming days. So as has been our practice with the previous vaccines, our role today is to think about the data, think about the process, and think about our preparation to provide a recommendation to Governor Lamont pending the EUA in terms of this vaccine being added to the vaccination program in Connecticut, always in a manner that looks ahead to the ACIP recommendations that will come down the line, and that we'll certainly share and consider as that comes along in the coming week. So that's the plan for our agenda today. We'll have lots of time, obviously, for item 4 and in the hour ahead. In terms of just committee activities, again, we, as mainly for the folks who are joining us from the public or the media, we note that our committee has been meeting monthly, at least monthly, throughout our duration. And after each meeting, we produce, with the help of our committee facilitator, Joe Albanese, detailed minutes of our conversations and our proceedings, which are shared with the group, to which additional comments can be added. And those are then finalized and shared with the advisory group. And we view those meeting minutes and these public conversations as really important additional sources by which our perspectives, our feedback is shared both publicly and shared with the advisory group. So that will remain our practice going forward, and those meeting minutes and transcripts are available to capture our committee discussions in addition to these video recordings. We'll be brief with respect to the vaccination program itself, but just again, another convention of giving some general updates in terms of where we are. At last report from the governor's office as of Thursday, we found ourselves nearing 900,000 doses of vaccines administered here in Connecticut, a performance overall that remains very favorable compared to other states. And you see those numbers with respect to the 75 plus and 65 to 74 population. The forecast is that Connecticut will receive another 130,000 first doses in this coming week. So between the updates, even since Thursday and the week ahead, it certainly seems likely that we'll pass 1 million doses administered, perhaps in the next week, or maybe very likely in the next week. So that work continues with respect to the vaccine distribution. With respect to vaccine allocation, of course, we've had the announcement from the governor earlier this week that the state would continue moving forward primarily in an age-based strategy beginning on Monday with individuals age 55 and above, with forecast dates for when future groups moving down the decades should expect eligibility to open, pending supply, pending additional developments between now and early May. So that will be the next sub-phase of vaccine allocation here in Connecticut, in addition to all those groups that are already eligible, as this slide shows, as well as the parallel program for educators for early childhood and pre-K through 12 educators and other individuals who work in those academic and childcare settings will have their vaccination programs beginning in this coming week, and hopes to move forward throughout the month of March with all of those individuals having access to first doses. So that was the brief update with respect to the committee, with respect to the advisor group. Thank you to all of you as well, I should mention, for all of the work. I know many of you have helped with the public forums and town halls and Q&A sessions that the communications subcommittee has been leading. So thank you. I've lost count of how many there have been, but I know many of you have spared some time on evenings and weekends to join those settings to help talk about the vaccines and the science. So thank you for that. We don't have any more that are in the queue right now, but I suspect those will continue, and we're grateful for your help in supporting that. So unless there are any big picture questions about the vaccination program and the advisor group, I think we'll turn things over to David next to take us through the safety and efficacy update. Here we go. David.
All right, thank you, Jason. So, we've had some opportunity to evaluate the efficacy and safety of these data in clinical studies, but now we're getting more and more real-world studies, and I think this is a great opportunity to review some of the data that's been published. So I'll run through a few studies that have been published in peer-reviewed literature, and then my last few slides will be based on some of the slides that were presented at the FDA VRBPAC meeting yesterday. So in terms of vaccine efficacy data, and this is actually effectiveness data, looking at the real-world implementation of this vaccine, this was a paper that was published last week in the New England Journal of Medicine that evaluated the effectiveness of the Pfizer-BioNTech mRNA vaccine rollout in Israel. This study looked at a large data source, so through this Clalit Health Services organization that is the largest in Israel, encompassing several million individuals enrolled in that organization, and collected data and analyzed vaccinated individuals versus unvaccinated individuals and looked at vaccine effectiveness. So for the next slide. So what this study did was between December 20th and February 1st, so this was the initial rollout of this vaccine in Israel. This analysis essentially compared those individuals who were vaccinated to those who were unvaccinated as controls that were matched based on demographic and clinical characteristics. And what they were seeking to evaluate was new infections with SARS-CoV-2. So they used several different outcomes, including symptomatic COVID-19, COVID-19 related hospitalizations, severe illness, and death. And they also included overall SARS-CoV-2 infection, which would encompass asymptomatic individuals. So that's an outcome that was generating a lot of interest, as we've discussed in previous meetings. And what they did looking at these different outcomes was calculate vaccine effectiveness, which is 1 minus the risk ratio using Kaplan-Meier analyses. So with regard to the outcomes, they looked at those several outcomes I mentioned on the previous slide in a few different groups, looking at several time periods post-vaccination. So they looked at individuals 2 to 3 weeks after the first vaccine, 3 to 4 weeks after the first vaccine, and then 7 days after the second dose of the Pfizer-BioNTech vaccine, which corresponded to 28 days after the first dose, given the 3-week interval between doses. So here's basically a description of the individuals included in this analysis. You can see a very large cohort consisting of 596,618 individuals who were vaccinated, and they were matched to the unvaccinated controls. Very sort of wide distribution. We didn't go into, in terms of age, we didn't really go into the entirety of the rollout of the vaccine in Israel, but they used sort of an age-based strategy with their initial rollout, focusing on older individuals and then subsequently moving down into younger cohorts, very similar to what's currently being used in Connecticut. In terms of medical comorbidities, we see that there's a sizable representation of these risk criteria, particularly obesity and smoking being the most common, but no significant differences between the vaccinated and unvaccinated groups with these different characteristics. And when we look at their outcomes, so the primary outcomes, we have the there's the Kaplan-Meier curves here on the right that look at cumulative incidence of infection. And then on the left here in table 2, we have the vaccine effectiveness data. And I won't go through every single category, but you can see on the bottom row, those who were 7 days after the second dose had a substantial vaccine effectiveness in terms of all the different outcomes. So symptomatic, hospitalization, severe disease, all well above or all above 90% or 85%. And with regard to the overall documented infections, that 92%, that includes asymptomatic individuals. So using sort of some calculations, they were able to identify what the vaccine effectiveness for preventing asymptomatic infection was, and that was 90%. So I think all very encouraging findings in terms of the real-world implementation of widespread vaccination in Israel. I'll point out a few key points. So a very large sample size. I think we're looking at including about 600,000 vaccinated and matched to 600,000 unvaccinated individuals. I think that's very impressive. I will also point out that Israel used a very rapid deployment of vaccines. So massive amount of vaccine coverage in a relatively short period of time. So kudos to their work there, and I think important to consider when we're looking at rolling out vaccines and vaccine effectiveness. But I think overall, very supportive data And then when it comes to looking at the variants, a lot of attention paid towards these emerging variants. And what this study did describe is, even though they didn't do specific analyses on effectiveness against specific variants, they did note that the B117 variant, which was the variant that was first identified as in the UK, had become the predominant variant in Israel during the study period. So as we start to think about variants emerging here in the US and in Connecticut, seeing that this was the predominant variant in the Israel population where we have this vaccine effectiveness data is encouraging. I mean, is there someone raising their hand? Jason, can you? Dr. Pettit, are you raising your hand? Do you have a question?
Yeah, David. I wondered if you had any insight into when they were screening people, how and if they excluded people with prior allergies. It's sort of beginning to be a common question from patients in the seminars and from constituents if they've included people with allergic issues because they have allergic issues themselves.
So are allergic issues that weren't included in vaccination? Is that what you're at, that were precluded from inclusion for vaccination? Is that what you're asking?
Oops. Did they try to screen all those people out, or could you have had atopic dermatitis, a little asthma, some allergy issues, and still be included?
So I didn't see any effort to exclude them. I can look a little bit deeper in that, but I don't believe there was specific mention of that as being an exclusion. So interesting question. I can circle back and take one more review of the study, but I didn't see that being specifically mentioned as an exclusion.
Yeah, I didn't see it in any of the writings on it either, so I wondered if they had talked about it in any other venues.
Yeah, I'll double check on that. If I find something, I'll circle back to the group.
And maybe just to help out, I think Israel early on had excluded people with severe allergies, but now they're starting to immunize them. They had a kind of a change in policy. Does that answer your question, Dr. Pettit?
Yeah, I mean, it's obviously the people who have allergies are wondering, were they included in the trial and did they do okay? So are they safe to take it is obviously essentially the question.
I'll take a closer look. I'll circle back to the group if it seems like that was specifically addressed in the study.
Right. In those weren't mild allergies. These were people with severe allergies in the beginning to anything were excluded in the beginning, but then they modified it.
All right. Thanks, Dr. Koh. All right. So can we move on to the next slide? So in terms of safety data, there have been a couple of studies released that describe sort of some follow-up to the early analyses that we discussed in the last in our last meeting. So this is the descriptive data from the VAERS system data set and VSafe that was between December 14th and January 13th, sort of the initial rollout, which mostly included Moderna first doses and Pfizer-BioNTech first doses, with a small group of Pfizer-BioNTech second doses included as well. And you can see the numbers are quite substantial, 13.8 million doses of the vaccines that were deployed during the first month of the vaccine rollout. Next slide. So here is a basic description of what was reported in VAERS. And you can see here when we're looking at sort of the overall cohort between the different groups, 5,000 Pfizer first doses, 1,300 Moderna first doses, and 100 Pfizer second doses. And I won't go through all these in too much detail, but you can see in terms of the adverse events that were reported, they really aligned quite nicely, I thought, with the clinical data from the phase 3 clinical trials for Pfizer, BioNTech, and Moderna studies. You can see the common symptoms: headache, fatigue, dizziness, chills, and nausea, occurring, ranging from about 14% to up to 25% with first doses of both the Moderna and the Pfizer, BioNTech, which aligns quite well with the data from the phase 3 trials. Next slide. And then there was a specific attention to anaphylaxis with both of the vaccines. And the overall picture, and on the left here, we have different tables that looked at so the overall proportion of individuals who developed each of the different adverse events in each of the different vaccines. You see predominantly injection site pain really dominated in terms of overall reports, and then less frequently the fatigue, myalgias, and chills, which you can see occurred generally quite early on after vaccination. Typically 1 to 2 days after both the first and second vaccine, much less frequently reported after the day 2 post-vaccination. So again, also very aligned with what was reported in the phase 3 trials. And then with regard to anaphylaxis in the total cohort, there were 4.5 cases per million of anaphylaxis. And just for some comparisons to flu, pneumococcal, and the zoster vaccine, you can see slightly above what we would expect to see with flu and pneumococcal vaccine, but below what's reported in terms of anaphylaxis after the zoster vaccine. And then the last or second to last piece of safety data that I'll report is what was presented at the VRPAC meeting yesterday. So this is data from the CDC vaccine safety data link system, and this is a collaborative effort between CDC and 9 healthcare organizations throughout the United States, which includes a large population, about 12 million people per year. And this is a system that was developed decades ago for studying other vaccines. And the goal is to collect clinical data from these 9 sites and conduct studies to identify rare events that are occurring, that may be occurring post-vaccination, and then compare them to what occurs in the unvaccinated group from these same organizations. And then they also do a sequential comparison of individuals pre and post-vaccination to identify whether rare events that occur following vaccination are occurring at a higher frequency in the vaccinated population versus what would be anticipated in a control population that is unvaccinated. And you can see with regard to the COVID-19 vaccines, they had a pretty substantial analysis that they performed, including over 600,000 individuals who received the first dose of the COVID vaccine and 200,000 who received the second dose of a COVID vaccine, and pretty evenly split between the Moderna and Pfizer vaccine. So you can go to the next slide. So what's performed is what they describe as a rapid cycle analysis. And so each week, this system basically analyzes the data from these 9 sites to identify what is the expected risk of different rare events based on what's seen in the population, and then how that compares to events that occur in the vaccinated population. So here on the left were some pre-specified endpoints that were evaluated comparing the unvaccinated group cohort versus the vaccinated cohort. And you can see, like for instance, acute myocardial infarction occurred in 23 individuals following a vaccine, and based on their analysis with the unvaccinated group, you would expect 26 events to occur. So they looked at, and these were several pre-specified endpoints that were evaluated specifically to see whether there was an increased risk of these events in post-vaccination. And what they found was in comparing the vaccinated group to the unvaccinated group, there were no statistically significant increases in risk for any of these pre-specified outcomes as of their analysis, which was their last analysis was February 13th. So about a couple weeks ago. And then on the right is a slightly different analysis where they look at rare outcomes through their sequential analysis, where they compare vaccinated individuals in the group prior to vaccine and post-vaccine to see if, based on their risk, they are seeing higher rates of events occurring post-vaccination. And what they found was for these outcomes, there were no significantly different, there's no specific statistically significant difference in terms of risk in post-vaccination. compared to the cohort pre-vaccination. And this is an important ongoing analysis that the CDC is performing, and they're going to get more granular in terms of their dose and product-specific analyses, and then they're going to do some interval analyses post-vaccination to see if these rare events are occurring at a higher frequency following vaccination than would be expected in the general population. So next slide. And then lastly, I'll conclude with some data on the pregnancy data. So this was data presented at VRPAC, sorry for the typo there. And it included pregnancy reports both through VSafe as well as VAERS. And essentially what was reported among pregnant women who were vaccinated with both vaccines. As expected, there were events that were reported. Generally, these were localized events and some systemic reactions that occurred post-pregnancy. So about 73% reported one of these localized or systemic reactions. And in terms of miscarriage, this was a key variable that the CDC wanted to follow closely. What they found was that it was the most frequently reported pregnancy-specific adverse event, and there were 29 cases, but that was compared to what the expected background rate of miscarriage would be, and found that this was not above what would be expected in the general population in terms of an overall background rate. So just a brief description of some of the pregnancy data. So I guess we can pause and see if there's any questions regarding any of the effectiveness or any of the adverse event data, safety data.
Yeah, David, one thing that wasn't mentioned that I know that Rick Martinello, I think, commented on at our last meeting, and a side effect that I personally experienced following Moderna vaccine and maybe to some extent following Pfizer, I'm not sure, that some people had sort of what appeared to be a delayed local hypersensitivity reaction at the injection site. So in my case, 9 days afterwards, I had maybe about a 3 or 4 centimeter wide round lesion surrounding the injection site that was red, itchy, slightly raised, lasted a couple of days, and then went away. I've since gotten my second dose. I'm second dose 7 days post that without any evidence of any local reaction like that happening this time, but it might be still a day or two early. But that was something that was mentioned. And at first there wasn't any clear instructions as to how somebody should handle that kind of a reaction with a second dose. But I think the experience is that people haven't experienced any adverse reactions more than expected with the second vaccine dose. But just wanted to point that one out since it was mentioned last time. And then of course I had noticed it, and I think for some people that might have been a concerning, what might happen if you get a second dose.
Thanks. I haven't seen any specific publication describing that in detail, though I believe it is addressed in some of the CDC guidance that individuals who develop that type of localized delayed type hypersensitivity, that it's being reported, and that individuals are tolerating their second vaccine. But do others have any comment on that? I'd be interested to hear either through experience with this vaccine or anything that they've encountered that's been published.
Hi, David. I do know the Yale Dermatology Group has submitted a manuscript describing a case series of patients who had delayed hypersensitivity reactions. As I understand it, all of the individuals, about a little over a dozen, all had similar issues to what Dr. Havler described, and they all self-resolved without incident.
Right. I'm actually, at David's suggestion, I actually contacted them, so I'm part of that case series. But I also read, I can't remember where I'd read it. It wasn't a formal publication, but there was a Harvard group that had seen the same type reactions, and they recommended going ahead and getting a second dose because in their experience, the second dose hadn't generated anything unusual that the first one was sort of heralding.
Dr. Pettit, did you have a comment?
No.
Okay, sorry, I saw your hand raised.
Yeah, I think, I think there's I know this is certainly the experience. A lot of this has come out in the press, but the NYU group has estimated 10%. There's a I know there's a JAMA article that mentions it from the CDC. So I think this is all probably pretty consistent in terms of what do you call it? This being a true, this delayed type hypersensitivity being a true mild or moderate side effect.
Thanks. If there are any other comments regarding the effectiveness and safety data, when we transition back to Jason, if you want to walk us through the next phase.
Absolutely. Thanks, David. And next slide here. So we will now turn to the J&J vaccine for brevity's sake in terms of names. And what we thought we'd do just before opening up for the discussion is just to briefly walk through some of the key results that have been reported and discussed over the last week. As you know, we shared the FDA and sponsor briefing documents with all of you in the subcommittee on Wednesday when they were released, about 200 pages worth of data. We also shared those presentation slides that were presented by both groups yesterday at VRBPAC as well. And some of you may have had a chance to watch some of those proceedings. So rather than duplicating that, I thought I just pulled out a handful of, I think, the key slides as they were presented verbatim by the FDA personnel in terms of how they interpreted the data, just some top line findings on both efficacy and safety that we can perhaps use to frame our conversation or add to it or to drill down deeper. And I will try to go through these relatively briskly since I think there's a lot of familiar ground here. So the vaccine candidate from J&J is a viral vector, recombinant replication incompetent viral vector. That's an important qualification. Adenovirus vector, a platform that's been used for other vaccine candidates, as well as the approved Ebola vaccine that Johnson & Johnson produces. A single dose regimen, as we are aware, and one that also comes with less arduous storage and handling requirements. I think it's up to 3 months at refrigerator temperatures is the particulars of this vaccine candidate. The key trial, obviously, there were many clinical trials preceding this pivotal phase 3 trial that's the focal point of this EUA submission. And there is a 2-dose vaccine trial, 2-dose regimen trial that's ongoing as well. But the trial here is that phase 3 trial of the 1-dose regimen, US, South Africa, and Latin America, 45,000 or so research subjects, ages 18 and above. And you can see the enrollment started in September and will be ongoing again for a 2-year duration overall. Primary endpoints for the trial were moderate to severe COVID-19. We have those case definitions available. I don't show them here, but we can bring them up if that's important to our conversation. Moderate to severe COVID-19 at either 14 or 18 days post-vaccination were the primary endpoints. And with respect to secondary endpoints, you see a range of them there, including severe critical COVID-19 cases of COVID-19 that require medical interventions, as well as deaths. The summary slides, again, as FDA presented them with respect to efficacy against that primary endpoint, moderate to severe critical COVID-19, was 66% at both 14 days after or 28 days after vaccination. In the United States, this is for the overall trial, for the United States subjects, that primary endpoint is 72% against moderate to severe COVID-19. When we look at that key secondary efficacy endpoint, the severe critical specifically, we see that efficacy of 85.4% at 28 days after administration of the vaccine. And then when we look further at that subset of severe critical cases that may require medical interventions, you see the numbers there, the 2 cases in the vaccine group, and then the larger numbers there in the placebo. And I'll show a little bit more of these in a moment. Also, by way of summary of FDA's view, they found that those efficacy outcomes were generally consistent across demographic subgroups. However, certainly as we look into the subgroups, as is true for this vaccine and others, the number of cases do get smaller, and the point estimates may need to be interpreted with caution. We see wider confidence intervals and with respect as we go deeper into the subgroups. And the one that particularly stood out was the lower efficacy among participants 60 years or older with comorbidities. FDA looked at those data and looked at, again, the number of cases, the fact that there was a shorter period of follow-up in that subgroup, given the way in which there was a staged enrollment. So there was only about 50 days of follow-up as opposed to that 2-month median follow-up that the overall research population had. So that's how they interpret perhaps why that point estimate is lower in that group, down somewhere around 43% rather than the broader group, but again, with a very substantial confidence interval around that point estimate. And overlapping confidence intervals with respect to the various countries where the trials took place. With respect, and I figured I'll just go through these and then we can have the free-flowing conversation. And I'm happy, of course, we have these slides and others that we might want to discuss. So here's that primary efficacy endpoint, that 66.1% finding here. And if we look just broadly at the over 60 and under 60 group, it's relatively consistent. If we look at the severe COVID-19 endpoint, there's that 85.4% vaccine efficacy at 28 days. You can see some differences there reflected by age with respect to the over 60 individuals with respect to that endpoint. There's the centrally confirmed, non-centrally confirmed case distinction that was brought up in VRBPAC, that positive COVID-19 PCR tests were also then sent to a central laboratory per protocol in Washington. And that's why there's something of over 90% alignment between those COVID-19 results. But that's why you see these two tables of centrally confirmed and non-centrally confirmed cases. As we look at that subset of the more severe cases, those that require medical intervention, hospitalization, ICU admission, mechanical ventilation, and or ECMO, you see that number at 28 days post-vaccination, where all of those cases that met that criteria following COVID-19 occurred in recipients of placebo, regardless of how that COVID-19, whether it was centrally confirmed or otherwise. And with respect to deaths, I don't show that slide here. With respect to deaths, there were 7 deaths, and all of which occurred among placebo recipients. No deaths related to COVID-19, I should say, among those who received the vaccine. That's the quick tour through efficacy. I'm sure we'll come back to it, but I just wanted to lay out some of the top line findings. With respect to safety, that was not, I think, a topic that the committee members saw a lot of reasons for concern about. I think that's a reasonable assessment. If others want to add to that, by all means. Here's the summary slide from FDA. Meets the expectations for the duration of follow-up. Reactogenicity, there absolutely were some local reactions, mostly pain, mostly mild and moderate, some systemic reactions, again, self-limited, some fatigue, but again, and headaches, mostly mild and moderate. We see there was a single adverse event of hypersensitivity, some cases, as I'll show in a minute, of hives reported in the vaccine group, and then a few, as I'll show here, some numerical imbalances of a few adverse events where the role, if any, of the vaccine could not be excluded as a contributing factor. So we see a 14 to 10 imbalance on thrombotic and embolic events there, where again, the vaccine can't be excluded as a contributing factor. Some cases of tinnitus and the cases of urticaria where there's an imbalance. And you can see how FDA has ascertained what we can think about the potential role of the vaccine or what needs to be further studied in that regard. So overall, before we turn to the group here, their overall assessment of benefits risks of this proposed EUA, they noted the reduced risk of symptomatic COVID-19 at least 14 days after vaccination, as well as that reduced risk of severe critical COVID-19 after 14 days, as well as the reduced risk of COVID-19 related hospitalizations and deaths, and that similarity across geographical regions, generally consistent efficacy across demographic groups and their assessment, and of course the advantages of a single dose regimen. Risks, the reactogenicity, local and systemic, is a risk. The hypersensitivity reactions, again, we return to. And then those few issues where the vaccine can't be ruled out as a contributing factor that I'm sure will continue to be studied and things we'll continue to talk about here in future meetings. With respect to the last slide, in terms of the data gaps and unknown risks that they noted for this vaccine, I think this is probably something would be true for all the vaccines we've discussed during our work together: duration of protection, the efficacy against asymptomatic infection or transmission, the role that new variants might play in diminishing efficacy, subpopulations such as those you see here, getting more data about safety and efficacy in those populations. Rare and long-term adverse event follow-ups will obviously continue to be observing, as we've seen, and then the possibility of evidence regarding the risk of vaccine-enhanced disease. So that is the 7 or 8 minute whirlwind tour, admittedly through the materials that I think you've been looking at and thinking about. And with that, I think we can open up the conversation for discussion about the vaccine. Again, both the question, I'm going to stop sharing so we can see each other a bit better. We can open up for any comments about the vaccine, about your overall impressions about the vaccine and its place here in our Connecticut vaccination program, and other issues which we may want to continue thinking about, talking about, and looking at as this vaccination moves forward and as we learn more about it. So we can open the floor to anyone who'd like to chime in on any of these topics around this vaccine candidate.
Jason, my 35,000-foot view is whether or not this committee would look at this data and make a recommendation to go forward, but to utilize it, try to utilize it mainly in a younger population, given some of the efficacy issues.
I definitely agree with that, Dr. Pettit.
Keith, I had trouble, I don't know, at least on my end, I had trouble hearing you. Can you repeat?
I said I agree with Dr. Pettit's assessment regarding the age-specific use of it. I mean, I know we're not going to approve it for age specificity, but just based on looking at the actual data itself, there's some advantages based on age.
Other perspectives on this topic. And again, we'll have guidance from ACIP that will help the state think about how to deploy the vaccine that will be coming in the coming days. So these are issues that I know will continue to be developed, things that we'll have a chance to continue to think about, if and when vaccination begins, if and when there's an EUA, and then this vaccine becomes introduced into the rollout. But other perspectives, we'd love to hear. Albert, I see you've unmuted.
Yeah, I think, well, first of all, this was this vaccine, I think from the larger perspective, I think there's nothing that, I could read that goes against the recommendations of, that would place caveats on the recommendations of VRBPAC or the FDA. This is a single dose vaccine. Efficacy is good, taking out the issues of, South Africa and the potential of variants. I think, again, the safety profile is really quite remarkable. And so this is definitely going to be a its use will be a public health success story. So the key question I think Representative Pettit is raising is about the robustness of the estimates on one specific age group or one strata of the age group. And that's, it's certainly the overall above 60 years of age. And in the other slides, in the addendum, over 65, it has pretty good equivalent performance compared to other or effectiveness compared to other. It's particularly those with comorbidities. And just to be clear, so is that the topic of discussion with the age group of 60 that's above 60 or 65 and with comorbidities, rather than just elderly?
I'll ask if that was Dr. Pettit's thought, and I don't want to speak for him in terms of how you were thinking. And I have a couple slides I can pull up about those subgroups while we talk here.
Just as to whether we have the ability in terms of getting the vaccine out to use this and try to send it to the clinics where the bulk of the people are going to be under 50 without significant comorbidities, and it may not show up for several weeks, so the timing may work for the rollout of the younger age groups. That was my only thought.
So let me just show, and I'll tee up, Albert, the slide you're thinking about. So this is the broader, this is a new slide here of the broader primary efficacy endpoints. And you see that there is this similarity certainly here, but to Albert's point, when you look at this next endpoint. And that's where this 42% that I referred to of the over 60s with a comorbidity, you see that the number of cases and the confidence intervals and how FDA interpreted what could or could not be inferred from that. But I think that is probably where you're thinking, Albert.
Yeah, and I think so. So I think separating out things, whether, what's the evidence for the elderly? And I think the evidence for the elderly is actually quite good. If you look at those with comorbidities, and here's the issue, you see that drop off. And there are two parts, and this was a discussion in VRPAC, but could it be because the confidence intervals are very wide, and the smaller, in the there are kind of low number of events in that group of over 60 with comorbidities. I think the things that are a little bit more worrisome, and so I would say that the issue is not just the elderly in general, it's the elderly with comorbidities. So then if you go to, in the sub-analysis, in the appendix, they did actually additional analyses. And what's a little bit concerning is that if it was just because of sample size, you wouldn't expect consistency across different endpoints. They found lower efficacy, not just for the primary endpoint of severe, moderate to severe, but they also saw it in severe disease and so forth. So I think at this point, it's really, that there's no smoking gun. You can't tell if it's because they didn't have enough events, or if it really does have lower efficacy in the elderly. That's going to require probably additional studies, which I know J&J is doing in that age group. But I would be, in that issue, and this is particularly in the nursing homes that we're concerned about. I think if there was a recommendation or advice or guidance to give to DPH, would be, at least in that population with over 60 with comorbidities, would occur to there may be better options for that. But again, doesn't necessarily give the message that everybody over 60 or 65 shouldn't receive this vaccine.
Rick.
Yeah, thank you very much. And I agree, with what Dr. Coe had just reviewed. And I think I had some initial reservations about this vaccine, thinking about it in reference to the RNA vaccines that we're currently using. But then, reviewing this data, thinking about, how the study was done, and in particular, this group over 60, something that, was really striking, and I think Albert just covered this very well, is if you go back to the slide that you were showing, Jason, in the 14-day group, essentially the vaccine effectiveness for those over 60 with comorbidities is really in the same ballpark as the rest of the data, coming in at 64.9%. And I think, to the point, which Albert covered very well, when we look at the 28-day data on the right-hand side, the numbers become smaller, the confidence intervals are wider. And while it's concerning that the average is lower, it's not clear to me that we can, really draw the conclusion that this is a deficit that the vaccine, has. Certainly a question that needs to be addressed as time goes on, but it's not clear to me that there's enough data here to really conclude that at this point. I think the other thing that's two other points. One is that if there are limitations in comparing this data to the Moderna and the Pfizer studies, but the overall vaccine effectiveness after the first dose is actually, it's greater. Whether it's truly greater or not is another question. And one thing also that I think came out, is that J&J is with a study, and I'm not sure what the timeline looks like for that study, but it's going to be very interesting, I think, to see what this data looks like and what the vaccine effectiveness is after receipt of the second dose in that separate study that's being done.
Thanks, Rick. Others? Jim.
Yes, I was going to bring up practical consideration for use of the vaccine, I know this is something that maybe, I guess, is going to be discussed by the ACIP as well, potentially on Monday, but especially as it relates to the elderly, and that is, as we've talked about before, nursing homes have turnover. So at the moment, we have very high vaccination rates. Looks like we're having a lot of success in reducing transmission there, but as the vaccinated population gets replaced with a potentially less vaccinated population, unless there's systematic efforts to keep them vaccinated, we could run into problems in the future. And this vaccine seems to be best positioned to sort of keep the long-term care facility population well vaccinated, because it can be stored in refrigerators on site, given on site, single dose. It seems like it's sort of the perfect thing for kind of maintaining high vaccination levels, whereas the other two vaccines at the moment are less so. I mean, you have to have a special effort and potentially have a special crew of people go around and offer vaccine periodically to all the newcomers and new staff and new patients. So interesting balance to try to strike there.
Maybe just for the sake of discussion, Jason, I think there are two important tables that were in the appendix of the VRPAC packet. It was slide 65 and 66. And it might be good just for people to take a look at that.
And Albert, they did present those slides yesterday? Oh, here we go.
Yeah.
I do have this here.
Yeah,
Hold on,
it's a packet that you had sent us.
Yep, I can jump over to this 65 and 66, and you should be able to see that now. So it's the PDF there. So here's 65. I don't know, Albert, if you want to walk through it, and then I'll flip to 66 when you're.
Yeah, so as Jim said, this is a trade-off, and again, the decisions are going to be made by the DPH and the governor. But so here they stratified the age rather than being 60 by 65. So you can see if you flip up one above or two above, this was the 2 pages up. Yeah.
There we go.
One more up. So you can see when they did the stratification with 60 and above, you get 32 and 42%, and they're not significant. And this is after 28 days after immunization. You go back down to the second one, two more down. So with this alternative, you can see that it's 44 and 44%. But then if you go one more slide down, one slide up.
There we go.
Yeah, this one. So you can see the. One more down. Apologies. So this one here, you can see with the severe and critical, and obviously these are small numbers, and you can see the intervals, confidence intervals have gotten bad, but you can see the consistency between the moderate and severe, and then the severe and critical, that they're both lower in the over 65. And they did make a note, and that was in a summary slide. I didn't see that in the packet by Johnson & Johnson, or the, in the packet, the original documents from Johnson & Johnson. But the concern here is that, there's some consistency. They did mention that when they increased the sample size, when they put more cases there, you could see there was an increase in the vaccine effectiveness, and if you go back up one slide, but you can see that, the numbers are low, but they're equally as low in those who are greater than 65 without any comorbidities. And you can see that, those are significant. So it's a little bit of a conundrum in, acknowledging Jim's, what Jim had just mentioned, that, certainly this would be the perfect vaccine to use in congregate settings, because of it's a single dose and it's easy and the cold chain. But I am a little bit concerned, and I think just that specific use in nursing homes, which is it's a vulnerable population, high mortality, high morbidity. If we come in with a vaccine that we're not sure about, would the better part of valor be to actually just keep with the more intensive vaccine that is working, and in that we're already witnessing decreases in hospitalizations and mortality. this is more a policy issue rather than maybe a recommendation for, I mean, this could be a suggestion or a consideration raised by our committee. This is certainly more of a policy issue that, DPH will have to, deal with.
Understood. Other comments? Oh, I'm sorry, Albert, please.
Yeah, no, I'm sorry. Does that make sense, Jason? That these were the two tables that were most concerning, I think.
Yes, and I'll note just deep into the day of the meeting, Peter Marks from FDA, the director of CBER, would ask the committee specifically if they had comments or advice about this particular slice of the subgroups and how to think about it. And there weren't a lot of comments, which Peter Marks took as a thought of saying it's an area for more interpretation, but not a yellow light or a red light. Other than the question is, when would we get more data? When might there be more data available, more time for follow-up, beyond the cutoffs that were used for the EUA, to which there weren't, clear answers from the manufacturer, but it was an area where it was clear that the need for more data to give some more precision in terms of these estimates, I thought were a high priority. That's what I took from that piece of the conversation yesterday. Other comments on this particular issue, broader issues about the vaccine, about the data, safety, efficacy? The floor is open. Let me unshare again so I can see you all a bit better.
This is Jim, just to follow up on Albert's comment. I think it's definitely worth noting that there's a potential trade-off, and until we get more data, we may want to have plans to try to sustain nursing home vaccination levels that focus on Moderna and Pfizer. I mean, recognizing that they're going to be more awkward to implement and could result in some delay in vaccination of some people that having a stock in your refrigerator wouldn't. But if the efficacy differences look like they're potentially big enough that it'd be better to take the less practical route in the short run. Until we're confident that we're not causing, that we're expedients wouldn't be costing us anything.
Well, Jim, the nursing home idea is not bad. We could check with DPH and Barbara Cass or some of the organizations, but perhaps not just the patients, perhaps there's significant turnover with the caregivers. And presumably most of the caregivers are under 60. So maybe that would be the perfect group to have it on hand when you have new CNAs, new aides, new people coming in, people who are working at several facilities, and you have that issue of cross-facility contamination. That may be helpful in that regard.
I think that's a great point, because there are two different groups we're focusing on there. Yeah.
Other thoughts? Yes, we'll certainly convey that this question, this implementation programmatic consideration, where there is this, uncertainty here that the state should think about. We'll make sure that issue is identified, particularly in, as I think Albert nicely quantified, not all older individuals, but particularly this group of older individuals with comorbidities where there is less clarity from the data. I think is a very fair way to think about what we can get and what we can't get from the data that we have to date.
Just one comment, and it's probably, I forgot exactly what was the follow-up period or the mean follow-up period for the Moderna and Pfizer, but I believe the Johnson & Johnson was shorter. It's, I think, about, 63% completed 2 months of follow-up. And so I do believe this is a little bit shorter than the other trials. That doesn't necessarily mean, I don't think that has necessarily implications for the, the approval or obviously for the EUA approval or its use. It's just, just guardrails need to be set down, or tracks to look at duration, especially since it's a one-dose vaccine. But certainly from its immunogenicity profile, it looks pretty good.
Right. No, Albert, as you were talking, I pulled up the slide there from that presentation. So FDA did require that median 2 months. as the floor before the EUA be submitted. So we see that overall. I don't have the actual overall slide here, but it is overall over 2 months. But that was one of those points that the FDA staff observed as we go down to this group that we've been thinking about today. That median follow-up in that subgroup is only 50 days, and that was part of their thought about why we might not see different kinds of case accrual patterns in the vaccinated versus unvaccinated arm. Not that it affirms any interpretation, but that was one of those differences they identified, since that is a pretty significant shorter follow-up in this particular group that we're thinking about here.
And I think, at least from what we're learning from Israel, and as David nicely presented, but also from England and Scotland, that it's not only effective against the B117 variant, but it's also one dose. And I believe there's actually additional information for the AstraZeneca that one dose, provides significant, protection, at least in the post-licensure studies and for in a smaller numbers. So, that's probably a lesser concern overall. I would expect, they're both adenovirus constructs and so forth. The immunogenicity profiles look, fairly similar.
I'm mindful of the clock here. Representative Pettit, I see you're unmuted, please.
The other question I had for people smarter than me to tell me about were the thrombotic and embolic events, whether those numbers were too small to be a major worry to us, or whether that's something we have to keep an eye on and wait for bigger numbers. I wasn't quite sure how to interpret the data that was presented on the thrombotic events.
I'll defer to others who might be closer to these questions.
At least, Representative Pettit, I believe they were equivalent. They were like 14 in one arm and 10 in the other, and the proportions were kind of equivalent. So nothing that kind of struck out. Unlike perhaps some of the data coming in for Pfizer and Moderna. But yeah, again, I guess it's in a much larger post-licensure surveillance. Some signals may pop up that weren't seen in the trials, but I don't think there were anything specifically worrisome. I'm not sure if other people thought differently.
Okay, thank you.
Yeah, and I agree with what Albert said. I didn't think too much of it, but I am curious to see if ACIP comments on it.
That's actually a good point from Rick. ACPI may take a very different take than what we're, than what we're saying, and it would be good to kind of regroup, or at least by email, to see what their input is. They may have very different thoughts on this.
Absolutely. No, and that's a good segue to think about next steps. So we're at that point that we've been in the previous patterns, where our immediate task before us over the weekend will be, once that EUA is issued, and I think that's fair to say it's when, not if, we will do what we've done in the past and share with you both the EUA statement and the FDA documents, the fact sheets. My sense is that the sense of the group is comfortable moving forward with this vaccine in Connecticut overall. And we will then, convey that recommendation pending your affirmation by email over the next day or so to the advisory group. We will note, as we have in the past, that recommendation, means that the vaccine should be administered in a manner consistent with how ACIP provides the granular recommendations. That's been the language we've used in the past. And then that will be an opportunity for us, when those recommendations are made public, again, in the next few days, early in the week, if not sooner. We'll share those with all of you. We can talk over email. We can continue this conversation, certainly, but between now and our next meeting. So that would be our plan for how to proceed. Is that agreeable to the committee?
Yes.
Yeah, I think so. Sounds good. Jason, just would you put, just to rate, would you put as a consideration the point that Representative Pettit said about, elderly? You could even say above 65 in comorbidities. Just
sure. No, we can certainly note that as an area that received, as it has, discussion among our group, as an area where there is perhaps a need for particular attention. We'll certainly communicate that formally. With that, David, do you have any other additional thoughts? Anything else we should think about in terms of next steps, or any comments to share?
No, I think we're on. We've got a plan for our upcoming discussion in the next few days via email. In terms of our next formal meeting, I don't have the date in front of me. I believe it's scheduled.
It's March 29th is when we are previously scheduled to have our next meeting.
Sounds good. So, as usual, we'll provide updates on how the vaccinations are proceeding at that meeting and effectiveness, efficacy, and safety data. But if others have any topics that would suggest for us to cover at future meetings, we can do that as well. Just share them with Jason and I, and we're happy to bring them forward.
Otherwise, I think we can thank you all for a good conversation. Thank you for the time on the weekend, and we will be in touch probably later tonight by email to finalize this piece of our work. So thank you very much for joining us, and we are adjourned. Have a good weekend.
All right, guys, thanks. Bye.
Thank you.
Yeah, thank you.